will explore the secondary and tertiary structures found within the FGFR2 receptor. The secondary structure of the protein is put together due to the interactions between amino acids in the sequence of the protein when the protein is being folded. The secondary structure of the FGFR2 receptor is composed of 3 alpha helices and 18 beta strands. The beta strands then come together in a lattice-like structure to form 2 beta barrels. The secondary structures then form the blueprint and scaffolding of the protein to give the FGFR2 protein its shape. This can be thought of as the framework of a house before the bricks and other materials are added on. In this schematic here, we can see the aforementioned alpha helices and the beta sheets. The FGFR2 receptor has 2 parts to it. Firstly, it has an area where the fibroblast growth factor can bind, and this area is circled in red, and the tyrosine kinase domain circled in blue. The tyrosine kinase domain is crucial in the downstream cascade which occurs to promote growth. Now that we have understood the secondary structure and the mechanism of action of the protein, let's unravel the truths of the tertiary structure. The tertiary structure is quintessential in allowing for the definitive shape and the lock and key mechanism associated with the receptor and the signaling molecule. The tertiary structure can be thought of as the materials required for the finished product of the house, including concrete, paint, bricks, and so forth. There are 2 main types of reactions that occur to form the tertiary structure of FGFR2 receptor. The first are disulfide bridges, which are the connections between 2 sulfur groups as seen here. And the second is glycosylation, the addition of carbohydrates to the protein. These reactions allow the protein to go from a rough framework seen here to a beautiful protein receptor ready to play an essential part in biological development.